Hello guys, welcome to my channel. Today we are going to discuss about the topic cells. This is very important topic in the perspective of exams like SSC, railways, state PSCs, etc. So I am discussing a detail about this topic cell, which is very important for these exams. All these points are very important. So, what are actually cells? Cells are basic building unit of all organisms. It is the fundamental structural and functional unit of all living organisms. So, it was discovered by scientist Robert Hooke in 1665. So, he actually discovered cells in cork slice using a primitive microscope. Primitive microscopes means an old type of microscope. So he discovered cells in cork slice. Then in year 1674, scientist Anton von Leeuwenhoek discovered free living cells. So living cells were discovered by Anton von Leeuwenhoek. He discovered free living cells in pond water. Then next, in 1831, Robert Brown discovered the nucleus. Nucleus is the center of the cell, and we can study more about this later. Next is protoplasm. Protoplasm was discovered by Purkinje in 1839. That is actually the fluid substance of the cell. Next, we can study about cell theory. Cell theory was actually formulated by Matthias Leiden, Theodor Schwann, and Rudolf Virchow. So in 1838, botanist Matthias Leiden is actually a botanist. He observed the plant cells and formulated the cell theory along with Theodor Schwann. Theodor Schwann is a zoologist. Theodor Schwann in 1839 observed the animal cells. And both Theodor Schwann and Matthias Leiden formulated the cell theory, which included 2 points. That is, cell is the basic unit of life and all living organisms are composed of cells. These are the 2 points of cell theory. Then in 1855, scientist Rudolf Virchow expanded the cell theory and added the point all cells arise from pre-existing cells. So all these points are known as the cell theory, and they were formulated by Matthias Leiden and Theodor Schwann, expanded by Rudolf Virchow. Next, we can look at the overview of a cell. This is the basic structure of the cell. So every cell has a cell membrane, which is the outermost membrane of the cell, and we have a liquid-like structure called cytoplasm, and we have a dense membranous structure at the center called nucleus, and we can have structures called organelles and ribosomes, which are non-membrane bound structures. So earlier we discussed about protoplasm. Protoplasm is actually everything inside the cell membrane. That is cytoplasm plus organelles plus nucleus. And cytoplasm is the semi-fluid matrix which occupies the volume of the cell. Now, based on the presence of a nuclear membrane, that is based on whether the nucleus is membrane bound structure, we can have 2 types of organisms, that is prokaryotes and eukaryotes. And prokaryotes, nuclear membrane is absent, while in eukaryotes, a nuclear membrane is present. So actually the cells have different shapes and size. We have the smallest cell, which is mycoplasma, and largest isolated single cell egg of an ostrich. Now we have 4 types of bacteria based on the shape of the cell. This is very important, and many questions are asked in terms of these 4 types of bacteria. So first one is bacillus. Bacillus means the shape of the cell is a rod shaped. rod-shaped cell is known as Bacillus bacteria. Next one is coccus. Coccus means the shape of the cell is spherical. Next, comma-shaped cells, bacteria is known as Vibrio, and spherical-shaped are known as Spirillum. So these four names, Bacillus, coccus, Vibrio, Spirillum, are very important. It was asked for several exams. You just need to know the names of the bacteria only and which shape it is. Next, we have prokaryotes. We have already mentioned that we have two types of organisms based on the presence of nuclear membrane. So, first one is prokaryotes, and questions are asked from prokaryotes section also. Nowadays, more questions are asked from prokaryotes along with eukaryotes. So, what are prokaryotes? They are actually organisms that lack nuclear membrane or membrane-bound nucleus. For example, bacteria, blue-green algae, mycoplasma, and PPLO. So these examples are important. That is bacteria, blue-green algae, mycoplasma, and PPLO. PPLO means pleuropneumonia-like organisms. So since nucleus is not membrane bound and continuous with cytoplasm, nucleus of prokaryotes is also called nucleoid. Then we have the parts of prokaryotes. First one is cell envelope. Cell envelope has three layers: glycocalyx, cell wall, and cell membrane. So, glycocalyx is the outermost layer of prokaryotic cell. It can be of two forms, either in the form of loose sheet called slime layer or in the form of a thick and tough layer called capsule. Next one is a cell wall. So, cell wall actually determines the shape of the cell and provides structural support to prevent the cell from bursting or collapsing. So, all prokaryotes contain cell wall except mycoplasma. In mycoplasma, cell wall is absent. Next one is cell membrane. Cell membrane is also known as plasma membrane. It is a selectively permeable membrane. Thus, selectively permits the entry and exit of various substances inside the cell. Next part of prokaryotes is mesosomes. So when we draw a rough picture of a prokaryotic cell, we have the outermost layer, which is glycocalyx. It is the outermost layer of the cell envelope. Then we have the cell wall and then a cell membrane. This is cell wall and this is cell membrane. Then we have mesosomes. Mesosomes means they are extensions of plasma membrane into the cell. So these extensions is known as mesosomes. And based on its structure, they can be vesicles, tubules, or lamellae. These are the three types of mesosomes based on its structure. And its major functions of mesosomes include cell wall formation, DNA replication and distribution to daughter cells, respiration, secretion, increased surface area of plasma membrane, and enzymatic content. So, in the case of cyanobacteria, these membranous extensions into the cytoplasm are called chromatophores. Chromatophores are the term given for mesosomes in cyanobacteria because it may contain pigments. Usually mesosomes does not contain pigments, but in case of cyanobacteria, it may contain pigments. So it is also called chromatophores. Then the next structure we have inside the prokaryotic cell is the ribosomes. And this is the cytoplasm. It is present in all cells. Ribosomes are the main site for protein synthesis inside the cell. And ribosomes of prokaryotes usually contains two subunits, that is 50S and 30S, and together 70S. And here S is actually Swedberg's unit, and we can study more about this when we look in detail about ribosomes. Now just see that the two subunits of ribosomes are 50S and 30S, and together it is 70S. So next, when a chain of ribosomes are attached to a single mRNA, it is called polyribosomes or polysomes. So that means polyribosomes means it is a chain of ribosomes attached to a single mRNA, also called polysomes. Then, and polysomes convert mRNA into proteins. Next, we have inclusion bodies. Inclusion bodies are present in cytoplasm, and it is actually used to store reserve materials. Reserve materials means, for example, phosphate granules, cyanophygean granules, glycogen granules, or gas vacuoles. The inclusion bodies are not actually bound by any membranes. They are just used to store reserve materials. So these are the main parts of prokaryotic cell, that we have cell envelope with three parts: mesosomes, ribosome, and inclusion bodies. And the nucleus of the prokaryotic cell is also known as nucleus. These are the important parts of prokaryotic cell. Next, we have a topic, bacteria. Nowadays, we can see many questions are asked from the portion of prokaryotic cells, especially bacteria and blue-green algae. So here I have provided a small description, important points about bacteria, mainly on the classifications of bacteria. First classification is gram-positive and gram-negative. So these classification is mainly based on the structure of cell envelope and response to staining procedure, which is just developed by the scientist Gram. So based on the response to that staining procedure, we have two types, that is gram-positive and gram-negative. So gram-positive means its cell wall is made up of thick layers of peptidoglycan, and also it retains the color of stain during the staining procedure. The color of the stain means it was crystal violet color. So during the staining procedure, gram-positive bacteria was able to retain the color of the stain. Then gram-negative bacteria was not able to retain the color of stain during the staining procedure. Also, it is made up of thin layers of peptidoglycan. So that is the main difference between gram-positive and gram-negative. Next classification is motile and non-motile bacteria. Basically, motility or motile bacteria means which are able to move. Those bacteria which are able to move are called motile, and those which are not motile means they are not able to move. They will be mainly attached to some tissues or any rocky substances, materials like that. So in the case of motile bacteria, movement of motile bacteria is due to structure called flagella. Flagella helps in the movement of this motile bacteria. And this flagella is actually a thin filamentous extensions from cell wall. If this is the cell wall, there will be a thin filamentous extension from the cell wall. This is known as flagella, and it has three parts, mainly filament, hook, and basal body. This is the basal body, this is the hook, and this is the filament. Non-motile bacteria have structures called pili and fimbriae. So pili is an elongated tubular structure made of special protein, while fimbri are small bristle-like fibers. This is the bristle-like fibers sprouting out of the cell, while pili is a long tubular structure from the cells. So both of these structures help non-motile bacteria to attach to host tissues or rocky surfaces. So that is the classification of motile and non-motile bacteria. Next type is eukaryotes. So eukaryotes are organisms with membrane-bound nucleus. We have examples: protists, plants, animals, and fungi. These examples are important. That is protists, plants, animals, and fungi. And these eukaryotes also contain membrane-bound distinct structures called organelles. Now, plant cells have a rigid cell wall and an organelle called plastids, both of which are absent in animal cells. While animal cells have an organelle called centrioles, which are absent in plant cells. And all other organelles are found in both animal and plant cells. But you need to study these three names, very important. That is cell walls and plastids and centrioles. That is cell walls and plastids are found in plant cells only, while centrioles are found in animal cells only. Next, we can look into the details of each cell organelles and each part of the cell. First one is cell membrane. So the cell membrane is actually composed of lipids, proteins, cholesterol, and carbohydrates. Mainly, it is composed of lipids and proteins. It may also contain cholesterol and carbohydrates. So here, lipids are actually arranged in bilayers. That means it has two layers of lipids arranged in the cell membrane. Then we have two types of proteins on cell membrane, peripheral and integral. Peripheral proteins lie on the surface of membrane, while integral proteins lie partially or totally buried in the membrane. So, in the year 1972, Singer and Nicholson made an improved model of cell membrane, which is also known as fluid mosaic model. So, this is the fluid mosaic model. And according to this model, the lipids of cell membrane have a quasi-fluid nature. This is the phospholipid molecules here shown in blue color, and they are arranged in bilayer. And here we have peripheral protein and integrated protein. Peripheral protein is lying on the surface of the membrane, while the integrated protein are either lying partially or totally buried in the membrane. So according to this model, the lipids of cell membrane have a quasi-fluid nature, and this enables the lateral movement of proteins within the overall bilayer of lipid. And this ability is known as fluidity. So this quasi-fluid nature of the lipid allows the protein to have a lateral movement across the cell membrane. Next, the main function of cell membrane is to transport molecules across it. So transport of molecules can be of two types: passive transport and active transport. So passive transports means molecules move across the membranes without any requirement of energy from higher concentration to lower concentration. So movement from higher to lower concentration does not require any energy. And so this type of transport is known as passive transport. Next, active transport means here molecules move from lower concentration to higher concentration. So this is an energy dependent transport process. So this is known as active transport. Next, passive transport of water is called osmosis. This is very important term. So actually this is the movement of water from higher concentration to lower concentration through a selectively permeable membrane, which is our plasma membrane or cell membrane. So the movement of water from higher concentration to lower concentration is known as osmosis. And plasma membranes are selectively permeable because it is selective in permitting the molecules to pass through it. Next part is cell wall. As I have already mentioned, this is not present in animal cells. Cell wall is actually a covering for the cell membrane, and it is usually present in fungi, protists, and some bacteria also. And apart from plant cells, it is found in fungi, protists, and some bacteria. And I have already discussed that it is not present in mycoplasma, and also some L-form bacteria, other plants, other bacteria, protists, fungi, and all. This cell wall is present. So it is actually a covering of cell membrane. It is a non-living rigid structure which forms the outer covering of plasma membrane. It is absent in animal cell. And the main component of plant cell is cellulose. This is very important. Main constituent of cell wall is cellulose. And main functions of cell wall include it gives shape to the cell, it protects cell from mechanical damage and infection, and helps in cell-to-cell interaction. It helps interaction between the cells, and it provides barrier to undesirable macromolecules. So I have already told that the main constituent of cell wall is cellulose. But in case of algae, it may contain galactans, mannans, calcium carbonate in addition to cellulose. And in case of plant cell, it may contain hemicellulose, proteins and pectins in addition to cellulose. Next, cell wall of a young plant cell is called a primary wall. And this primary wall is able to grow. It is capable of growth. But when plant matures, this primary cell wall diminishes and a secondary wall is formed. Next, middle lamellar layer. So, middle lamellar layer is actually used to connect or held together to hold the neighboring cells together. So, if these are some of the cells, they are held together by a middle lamellar layer. And this middle lamellar layer is made of calcium pectate. Then next we have plasmodesmata. So plasmodesmata actually used to connect neighboring cells. Cytoplasm of neighboring cells. Cytoplasm of neighboring cells are actually connected using plasmodesmata, and it traverses the cell wall and middle lamellar layer to connect the cytoplasm of two cells. Next, we have endomembrane system. Actually, endomembrane system is a group of membranous organelles whose functions are coordinated with each other. And this includes, that is, endomembrane system includes 4 organelles, and you need to study all these 4 names which are included in endomembrane system. That is endoplasmic reticulum, Golgi complex, lysosomes, and vacuoles. So questions may ask like those organelles which are not included in the endomembrane system or which are included in the endomembrane system. So you need to study about these 4 names. There is endoplasmic reticulum, Golgi complex, lysosomes, and vacuoles. So first endomembrane system is endoplasmic reticulum. So this is the basic structure of an endoplasmic reticulum. So it is also known as ER. So ER is actually a network of tiny tubular structures scattered in the cytoplasm. And its main function is to transport materials between cytoplasm and nucleus. So here ER actually divides the intracellular space into two compartments, luminal space and extraluminal space. The luminal space means the space inside the ER. This is the space inside the ER. And extraluminal means which is in the cytoplasm, that is outside the ER, or the space inside the cytoplasm is called extraluminal. And ER may have or may not have ribosomes, and based on that we have two types of ER, RER and SCR. Rough endoplasmic reticulum or RER actually has ribosomes on it, and it is continuous with nuclear membrane. Here we can see it is continuous with the nuclear membrane, and it helps in protein synthesis. It actually contains the ribosomes. Next, we have smooth endoplasmic reticulum or SCR. SCR does not have ribosomes on it. It helps in lipid synthesis, and in animal cells, it also helps in steroidal hormone synthesis and all. But its main duty is to synthesize lipids. Next, this SCR can help in the process of building cell membrane, and this process is known as membrane biogenesis. So this is very important. Question may ask which type of organelle helps in membrane biogenesis, and it is SCR. Next, we have Golgi apparatus or Golgi bodies, Golgi complex, etc. So Golgi apparatus was first discovered by Camillo Golgi in the year 1898. So this Golgi apparatus actually consists of many flat disc-shaped sacs stacked parallel to each other. They are known as cisternae. So all these disc-shaped structures are known as cisternae. And its main function is packaging of materials which are to be delivered either to intracellular targets or secreted outside the cells. So here, Golgi apparatus actually has two phases, cis phase and trans phase. So cis phase receives vesicles, that is, cis phase is the convex side of Golgi apparatus. This is the convex side. It is also known as forming phase. And this phase receives unmodified proteins in the form of vesicles from the endoplasmic reticulum. So cis phase phases ER. That is, ER produces proteins and lipids, and this will be unmodified. So cis phase of Golgi apparatus receives this incoming vesicles from ER and modifies the proteins and lipids. So this is the receiving side of the Golgi apparatus. Next is trans phase. This is the concave side of the Golgi apparatus, and it is also known as maturing phase. And it is the exiting phase from which vesicles leave the Golgi bodies. So trans phase actually phases the cytoplasm. So the unmodified proteins in the form of vesicles from the ER and the cis phase, it gets modified inside the Golgi bodies and exit through trans phase. Actually, proteins and lipids from ER are modified to glycoproteins and glycolipids inside the Golgi bodies and exits through trans phase. Next, we have lysosomes. So lysosomes are actually membrane-bound vesicular structures formed by the process of packaging in the Golgi apparatus. So Golgi apparatus exits some vesicles, that is modified proteins and lipids from the trans phase of the Golgi apparatus. So those vesicles are membrane-bound vesicular structures formed by the process of packaging in the Golgi apparatus, and these are known as lysosomes. And they are rich in all types of hydrolytic enzymes like lipases, proteases, carbohydrates, which are active at acidic pH. And these enzymes are capable of digesting carbohydrates, proteins, lipids, and nucleic acids. And lysosomes actually keep the cell clean by digesting any foreign material and as well as worn out cell organelles. So when cells gets damaged, lysosomes may burst and enzymes digest their own cell. So lysosomes are also known as suicide bags of a cell. So it's very important which organelle is known as suicide bag of the cell. It is lysosomes because it contains so many digestive enzymes. So when the cell gets damaged, lysosomes burst and use these digestive enzymes to digest their own cell. So that is why lysosomes are also known as suicide bags of a cell. Next is vacuoles. So actually in the case of plant cells, 90% of its intracellular space is covered by vacuoles. But in the case of animal cells, it contains only small vacuoles, but in animal cells, it occupies 90% of this intracellular space. So vacuoles are membrane-bound space found in cytoplasm and it is used for storage. And different organisms have different materials in vacuoles. And the membrane of the vacuole is called tonoplast. And this helps to transport ions and other materials against concentration gradients. That is, it works against concentration gradients. Name is tonoplast. Next, I have already mentioned different organisms store different materials in vacuoles. In case of plant cells, vacuoles contain water and sap. Plant sap. Plant sap includes amino acids, sugar, organic acids, proteins, etc. And some organisms store excretory product and other materials not useful for the cell. So, and it expels the waste using vacuoles. So, vacuoles helps in excretion also. Now, in the case of protist, these vacuoles are known as food vacuoles because food vacuoles contain food item it has consumed, the protist has consumed. So it is also known as food vacuoles. Next, in the case of amoeba, the vacuoles are also known as contractile vacuole because it helps in osmoregulation and also excretion. So osmoregulation means the process of maintaining the balance of water inside the cell. So here the contractile vacuole helps in osmoregulation, that is balancing the water content inside the cell and also excretion. So this term is very important, that is amoeba contains contractile vacuole. Next, the organelles associated with endomembrane system are completed. Next, we can discuss other cell organelles and cell parts. So first thing is mitochondria. So mitochondria are actually membrane-bound cell organelles that generate most of the chemical energy, which is needed for the cell for various biochemical reactions. The most of the cellular energy is actually produced by mitochondria. So this is the basic structure of a mitochondria. It is double membrane. It has an outer membrane and an inner membrane. Its inner compartment is actually filled with dense homogeneous substance called matrix. The outer membrane is continuous limiting boundary of the organelle, while the inner membrane forms foldings called cristae. And towards the matrix. So inner membrane has foldings into the matrix, while outer membrane is continuous. And mitochondria is actually the main site of aerobic respiration. And it produces cellular energy in the form of ATP. ATP means adenosine triphosphate. And this energy is required for various chemical activities needed for life. Hence, mitochondria is called powerhouse of the cell. And this is a very, very important question, a repeat question for various exams. The powerhouse of the cell and the energy, cellular energy is produced in the form of ATP. So, ATP is known as energy currency of the cell. So, my very important question, which organelle is known as powerhouse of the cell? It is mitochondria. And energy currency of the cell, ATP, is known as energy currency of the cell. Next one is ribosomes. So, ribosomes are actually non-membrane bound organelle. It does not have any membrane. And it was first observed by scientist George Palade in the year 1853, sorry, 1953. It is a non-membrane bound cell organ. It is a granular structure actually composed of RNA, that is ribonucleic acid, and proteins. And it is not surrounded by any membrane. And I have already discussed that ribosome contains two subunits. And in the case of eukaryotic cells, that is 60S and 40S. And in prokaryotic, it is 50S and 30S, together 70S. In case of eukaryotic, 60S and 40S, and together it is 80S. So actually here S means it is Swedberg's unit. That is, it's a unit of sedimentation coefficient. Need not study more detailed about this. You just need to know that this unit gives an indirect measurement of density and size of ribosomes. So 60S and 40S and all represent the measurement of density and size. You don't need to know any more details about it for these exams. Just know that the subunits of eukaryotic and prokaryotic, that is eukaryotic, it is 60S and 40S, together 80S, and in prokaryotic, together it is 70S, 50S, and 30S are the subunits. Next is microbodies. Microbodies are actually membrane-bound vesicles containing various enzymes, and it is present in both plant and animal cells. These are not actually asked, it's not asked in any exams, but I just mentioned for it for an extra information. It is actually membrane-bound vesicles containing various enzymes, and it is present in both plant and animal cells. Next, we have plastids. So plastids are usually found in plants and euglenoids. And they contain specific pigments. So it imparts specific colors to plants. And based on which pigments, which type of pigments are found in different plastids are divided into three types. First one is chloroplast. Chloroplast, chromoplast, and leucoplast are the classification. First one is chloroplast. Chloroplast contain chlorophyll, which imparts green color to plants, which is very essential for photosynthesis. So chlorophyll provides green color to plants. Next, chromoplast. Chromoplast contain carotenoid pigments. That is pigments like carotene, xanthophylls, etc. And they impart yellow, orange, or red color to the plants. Next, we have leucoplast. Leucoplast does not have any pigment. It is actually colorless plastids, but it is used to store nutrients. And based on which type of nutrients it stores, we have three types of leucoplast. Amyloplast is the first one. So amyloplast actually store carbohydrates or starch, and this is usually found in plants like potato. Next is elaioplast. Elaioplast store fats and oils. The next one is eleuroplast. Eleuroplast store proteins. So these are the three types of leucoplast, and based on which type of nutrients it stores, these are the names of the classifications. Next, we have already talked about chloroplast. It contains the chlorophyll pigment, which imparts green color to the plants. So this is the basic structure of a chloroplast. It is actually a double membrane organelle. It is found in mesophyll cells of leaves. Now, we have two membrane, outer membrane and inner membrane. And the space inside the inner membrane is called stroma. Stroma contains a number of organized flattened membranous sacs called thylakoids. So these are the various disc-shaped structures arranged in stacks, and they each disc is known as thylakoids. And chlorophyll pigments are actually present in the thylakoid. Next, stacks of thylakoids form a grana. And thylakoids of different grana are connected using flat membrane tubules called stroma lamellae. Then the space enclosed by the membrane of thylakoid is called lumen. That is the space inside the membrane of thylakoid. It is called lumen. Then the stroma actually contains enzymes required for synthesis of carbohydrates and proteins. So this is the basic structure of a chloroplast. Next one is cytoskeleton. This is also not very important from the exam point of view. There is actually microtubules or microfilaments present in cytoplasm. That is, it is a collection of microtubules, microfilaments present in the cytoplasm. And this network of filament is proteinaceous structures. Functions include mechanical support, and it also maintains the shape of the cell. So these are the functions of cytoskeleton. This mechanical support maintains the shape of the cell, and motility. It is also the function of cytoskeleton. This is also not very much important from the exam point of view, but I just shared an extra information. Next one is cilia and flagella. So cilia and flagella are actually outgrowth from cell membrane, which causes movement of cell. That is, cilia is actually small hair-like outgrowth from cell membrane, which causes movement of cell or surrounding fluid, while flagella is longer than cilia but have the same function, that is cell movement. And this is the basic structure of a cilia or a flagella. This is the core of the cilia, that is also known as axonate. Usually, cilia or flagella are covered with plasma membrane, and the core of the cilia or flagella is known as axonate. This is the basic structure of axonate. Axonate contains microtubules running parallel to long axis. This tubule shown in blue color is known as microtubules, and we have 9 doublets of peripheral microtubules. That is, doublets means 2. I have shown only 2 pairs here. Actually, there are 9 pairs. They are known as doublets, peripheral doublets, peripheral microtubules. And we have 9 of them. And we have a pair of centrally located microtubules, and these are connected by bridges, and they are enclosed by a central sheath. And this central sheath is connected to one of the tubules of each peripheral doublets by using a radial spoke. This is a radial spoke. And the peripheral doublets are interconnected using linkers. This is the basic structure of an axon. Next, we have centrosome. Centrosome is actually an organelle containing 2 cylindrical structures called centrioles perpendicular to each other. So it actually helps in regulating the cell motility, and it facilitates the organization of the spindle poles during mitosis. So these terms are actually connected with cell division. So we can have a better idea when we discuss the topic cell division. I will have a separate session on cell division where you can understand more about centrosome. So now you just understand the basics, that all that is it is helpful during the cell division phase. It helps in the organization of the spindle poles during mitosis. And this centrosome contains 2 cylindrical structures called centrioles, and these centrioles are perpendicular to each other. Centrosome is also known as microtubule organizing center of the cell. It contains proteins for the formation of microtubule. So this is the basic information of centrosome. We can study more details about it during cell division session. Next is the nucleus. So nucleus is a very important part. It was discovered by Robert Brown. I have already discussed. It is in 1831. This is the basic structure of a nucleus. Contains nuclear membrane and nuclear pore, nucleolus, and a chromatin material or DNA. It is a thread-like structure, and we have a liquid matrix inside the nucleus called nucleoplast. So this is the basic structure of a nucleus. So nucleus is responsible for regulating almost all forms of cellular activities inside the cell. So it is also known as the brain of the cell. Now the membrane of the nucleus has it is double membrane. It has an outer membrane and an inner membrane. And it have nuclear pores which are formed by the fusion of two membranes. Outer membrane actually remains continuous with ER and bare ribosomes. We have already discussed in the case of ER that ER is continuous with the nuclear membrane and it has ribosomes. Nuclear pores. Nuclear pores are actually used as a passage for RNA and protein between nucleus and cytoplasm. So it is actually passage of protein between nucleus and cytoplasm. Then we have nucleoplasm, which contains nucleolus and chromatin. And nucleolus is actually not a membrane-bound structure. It is actually continuous with nucleoplasm. It does not contain a membrane, a well-defined membrane in it all. That is continuous with nucleoplasm. Nucleolus is actually the site for active ribosomal RNA synthesis. Next, we have this chromatin material. During different stages of cell divisions, chromatin material is known as chromosomes. So actually, chromatin material consists of DNA, RNA, and histones. Histones are actually a type of proteins. So this is the basic structure of chromatin, but during cell division, it is known as chromosomes, and chromosomes contains centromere and kinetochores. This is the disc-shaped kinetochore we have, and these are the arms of centromere. So centromere is a primary constriction which holds two chromatids of a chromosome. And kinetochore is a disc-shaped structure. Now, based on the position of the centromere, we have four types of centromeres, chromosomes. That is metacentric, subcentric, acrocentric, and telocentric. Metacentric means the centromere will be at the middle. That is, we have two equal arms of chromosomes. Now, subcentric means the centromere is slightly away from it, that we have a short and a long arm centromeres. Next, we have acrocenter, where centromere is situated close to one end. So we have extremely short arm and very long arms. Then we have telocentric, where we have a terminal centromere. So, and then some have secondary constrictions at a constant location in the form of small fragments. And this constrictions are known as satellite. So this is the basics about centromere, chromatin material, and chromosomes and all. We can have a detailed discussion when we look into the topic cell division, which I will create a separate session. I have prepared a detailed notes based on the topic cell, which you can get at just ₹20. You heard it right, it's so cheap. For such a cheap rate, you are getting a detailed preparation material along with 85 plus questions based on topic cell. The questions include both previous questions and practice questions. This material will be useful for all aspirants of SSC, RRB, NTPC, State PSC, and even NEET exams. Just send a mail to mail ID provided in the description with subject cell and get the notes containing 25 plus pages at just ₹20. Please like, share and subscribe my channel. Press the bell icon to receive notifications and comment your reviews.